I'm Dr. Chris Masterjohn of chrismasterjohnphd.com, and I'm here with Alex Leaf.
Hey.
And you are listening to episode 54 of Mastering Nutrition, where we talk about pantothenic acid, part 1, what it is and why we need it.
This is Mastering Nutrition with Chris Masterjohn. Take control of your health, master the science, and apply it like a pro. Are you ready?
Vitamin B5, or pantothenic acid, is important to your energy. It's important to your mood. It's important to your emotions. It is important to your digestive system. It is important to your ability to go through life with the ability to move gracefully and normally without experiencing cramps, without experiencing cramps abdominally, where they're driven by your digestive distress, without farting too much. And in the worst case scenarios, it might really mess with your neurological system, causing tingling in the hands and feet, or an inability to walk normally, and so on. In fact, one of the key signs of vitamin B5 deficiency is insomnia. Despite the fact that most of these things are common problems, the conventional dogma is that pantothenic acid deficiency doesn't exist naturally. Pantothenic acid is named after the Greek word pantos, which means everywhere, everything. It's in everything. It's everywhere. Therefore, you will not become deficient in it. In this two-part series on pantothenic acid, Alex Leaf and I deconstruct this idea that you will never run deficient in pantothenic acid. And we show why, in fact, we should not regard pantothenic acid as the vitamin that you'll never be deficient in. We should regard it as the mystery vitamin, the vitamin where we have no idea what the prevalence of deficiency is, and where we have lots of good reasons to think that many people would need more. In this series, I focus on the basic science of the nutrient, how much we need, how to use testing to determine whether we have enough, and how to get it from food. Alex focuses on supplements. In part 1, we focus on the basic science of what it is and what it does in the body, why we need it. Part 2 will be on the extremely practical stuff of how much do we need, how do we get it from food, how do we use lab testing to know whether we have enough, and what are the health outcomes of supplements. This episode covers the biochemistry of what it does, its role in breaking down food molecules for energy, in synthesizing things like fatty acids, ketones, and cholesterol, and sex hormones, and other steroid hormones that regulate your blood pressure and your electrolyte balance and your blood sugar. Its role in controlling neuropeptides that control things like appetite, libido, and the graying of hair. Its role in helping you utilize iron properly so that it goes towards good things and doesn't cause damage. Its role in making acetylcholine, needed for learning, memory, and sustained focused attention, or to allowing you to dispose of protein without ammonia toxicity, or to making the mucus that lubricates the ears, eyes, nose, mouth, vagina, penis, and internal organs. Its role in making melatonin to help you sleep at night. Its role in folate metabolism and the synthesis of glycine, ATP, components of DNA and RNA, and why you need 20% more pantothenic acid when you burn fat than when you burn carbs, but why, if you're very deficient or if you have an impairment in your metabolism of B5, a high-fat diet could have devastating consequences that are far greater than that 20% figure would suggest. We talk about how it's absorbed and distributed in the body, and how in 2015, a paper in Nature Biological Chemistry completely overturned, or at least provided a foundational evidence for potentially completely overturning much of the last centuries of work on how it's absorbed and distributed and circulates in the body. We guesstimate that we can probably store about 6 years worth of pantothenic acid in our bodies. And yet it might take as little as 9 weeks of eating a very poor diet to have serious consequences develop. We cover why needs increase in adolescence, pregnancy, and lactation. And finally, we cover why food forms of vitamin B5 may well be superior to any forms available in supplements, and why this is almost certainly true in a very meaningful way for people with a rare genetic defect in the ability to activate vitamin B5, but how we can learn from that extreme example to make a compelling argument that getting vitamin B5 from food is the ideal way to get it. And in fact, because we know so little, many of us may have the same problem activating B5 from supplements, just to a much lesser degree than the well-established severe genetic defects. And yet, there are supplement trials that use 6,000 times the dose that you would usually get from food and have proven to be completely safe and utilized with stunningly high proportions of those doses. This is part 1 of a 2-part series. Since it is episode 64 of Mastering Nutrition, you will be able to find the show notes now and forever at chrismasterjohnphd.com/64. Part 2 will be published 2 weeks from today. If you want it right now, you can sign up for the CMJ Masterpass at chrismasterjohnphd.com/masterpass/mastering nutrition, or use the link in the episode description in order to get a lifetime discount of 10% off. You can also go there to find a transcript of this episode. If you want all the episodes of the series on managing nutritional status for each nutrient, then you can find those at chrismasterjohnphd.com/marker #episodes. Or just go to chrismasterjohnphd.com/marker and scroll down to the episode section, or just use the clickable link in the description of this episode. If you want to get notified by email when new episodes come out, go to chrismasterjohnphd.com/mastering nutrition. With all that said, let's dive deep into our discussion of the science of vitamin B5 right after this word from my sponsors. This episode is brought to you by Ancestral Supplements. Traditional peoples, Native Americans, and early ancestral healers believed that eating the organs from a healthy animal would strengthen and support the health of the corresponding organ of the individual. For example, the traditional way of treating a person with a weak heart was to feed the person the heart of a healthy animal. Modern science makes sense of this. Heart is uniquely rich in coenzyme Q10, which supports heart health. The importance of eating organs, though, is much broader than simply matching the organ you eat to the organ you want to nourish. For example, natives of the Arctic had very limited access to plant foods and got their vitamin C from adrenal glands. And vitamin C is important to far more parts of your body than simply your adrenals. In his epic work, Nutrition and Physical Degeneration, Weston Price recorded a story of natives who cured blindness using eyeballs, which are very rich in vitamin A. But now that we understand vitamin A, we know that we can get even more vitamin A by eating liver, making liver good for your eyes. Our ancestors made liberal use of organ meats both to be economical and to utilize their healing and nourishing properties. Animals in the wild do the same. Weston Price had also recorded a story of how the zoos in his era were capturing lions, tigers, and leopards, oh my, only to watch them become infertile in captivity. Researchers then observed what the lions did when they killed zebras in the wild. What they did was they went straight for the organs and bone marrow, leaving the muscle meat behind for the birds. But even the birds took what they could of the organs and bone marrow. Price reported that once the zookeepers started feeding the animals organ meats, boom, their fertility returned. The problem I often encounter, though, is that many people just don't like eating organ meats. Let's face it, if you weren't raised on them, it can be very hard to acquire a taste for them. That is where Ancestral comes in. Ancestral Supplements has a nose-to-tail product line of grass-fed liver, organ meats, living collagen, bone marrow, and more, all in the convenience of a gelatin capsule. For more information or to buy any of their products, go to ancestralsupplements.com. Ancestral Supplements, putting back in what the modern world has left out. This episode is brought to you by Ample. Ample is incredible. It's a meal in a bottle that takes a total of 2 minutes to prepare, consume, and clean up. 2 minutes. I'm not kidding. Now, I know what you're thinking. Anything that quick just has to be made of synthetic ingredients that you'd have a hard time pronouncing and wouldn't want to put into your body. But it's not. Ample is made entirely from natural ingredients and designed to provide an optimal balance between protein, fat, and carbs, as well as all the vitamins and minerals that you'd need in a single meal. There's no question that it's always best to sit down and take your time eating a home-cooked meal from fresh ingredients. But let's face it, oftentimes we just don't have time for that. If you live a busy life like I do and your goal is to get things done, you need quality fuel that you can get into your system quickly. Here's a great example where Ample is perfect for me. When I shoot videos, it takes hours to set up and break down all of my equipment. So I try to get as many videos shot in a day as possible. This prevents wasting a lot of time on setup and helps me conserve big blocks of time outside of shooting videos to get into a flow state where I can research something to my heart's content and spend all the time I need thinking about it creatively and analytically. But if I spend hours dealing with recording equipment plus hours spent preparing food, eating it, and cleaning up, there's no time left over to actually shoot any videos. So on recording days, I use Ample to maximize efficiency and focus on getting things done. Ample comes in 3 versions: original, keto, and vegan. And each version comes in 2 portion sizes, 400 calorie and 600 calorie. The 600 calorie original version gives me 37 grams of protein from a mix of grass-fed whey and collagen, which promotes satiety and flips my brain on. Its fat comes from coconut oil and macadamia nut oil. I like these oils because they're low in polyunsaturated fatty acids, or PUFAs, oils that promote aging and are usually loaded into the processed foods that most people eat when they need something on the go. The coconut oil provides some medium-chain fats to keep my energy levels up, too. The carbs, the vitamins, and the minerals all come exclusively from food sources like sweet potatoes, bananas, cocoa powder, wheat and barley grass, and chlorella. It's full of natural prebiotic fibers and probiotics to promote a healthy microbiome, and the gentle sweetness comes from a mix of honey, monk fruit, and stevia. I just mix it with water, drink it, rinse out the bottle, and boom, 2 minutes in, and I'm fully fueled and ready to face the next phase of the day. I first came across Ample when I met its founder and CEO, Connor Young, at PaleoFX a few years ago. Connor inspired me with his vision for Ample, which I anticipate will be much more than a meal in a bottle in the near future. I've become an official advisor to Ample, and I'll be helping Ample design scientific research that will lead both to an ever-improving Ample and, I hope, meaningful contributions to our understanding of how to use nutrition to help people be healthier and happier and perform better at the challenges that they care most about. As a listener to the Mastering Nutrition podcast, I've also worked out a special deal for you. If you use the discount code CHRIS15, you'll get 15% off your first order of Ample. To get your discount, go to amplemeal.com. That's amplemeal.com, 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 and use the code CHRIS15 at checkout. Imagine that you feel fatigued. You don't feel like you have any energy. People are looking at you, and the thing that they think is you just don't have any enthusiasm anymore. But now imagine that you start arguing with other people more. You are kind of down in the dumps. You're acting childish, and you can even observe in yourself that you're acting childish. Sometimes you just want to spend the entire day in bed. As things get worse, you start developing numbness and tingling in the hands and feet. You start snapping your hand, wringing it, trying to restore normal sensation. You start stomping your foot on the ground, trying to get the tingling to go away. Your pulse starts beating not super fast, but a lot higher than you would expect it to when you do even minor things, moderate exercise, or even just getting out of bed. you cannot fall asleep. You start developing cramps in random places in your body, but especially in your stomach. Sometimes you get nauseous. You get, occasionally you might vomit, and you've been farting a lot. Maybe your feet start burning, or maybe you start walking funny. Like you feel like you need to pick your foot up further than you should have to in order to keep walking. What has happened to you? What has happened to you is that you have been the subject in a controlled experiment inducing pantothenic acid deficiency. But because it's so hard to induce a pantothenic acid deficiency, they have given you an antagonist of the vitamin. In fact, they had to increase the dose of the antagonist after a few weeks of taking it because the symptoms started to go away. Although we have a well-characterized syndrome of vitamin B5 or pantothenic acid deficiency, we have the belief that it just doesn't occur naturally. In fact, pantothenic acid is named after the Greek word pantos, which means everything, everywhere. Pantothenic acid is everywhere. Pantothenic acid is in everything. Therefore, it's impossible for you to develop a natural deficiency. Because of this belief, pantothenic acid is what I would call the mystery vitamin. Of all the B vitamins, it's the one that we understand the least, the one that we know the least about. And I don't mean that we don't know what it does in the body. We know a lot about what it does in the body, and I'll be able to explain a lot of that today. But what we don't really understand is how much of it do we need for optimal health? What are the things that make us need more? How do we smack numbers on this and decide what to eat and how to look for whether we don't have enough? In fact, in 1998, when the Institute of Medicine's Food and Nutrition Board made the DRI, which is the dietary reference intake, usually what they want to do to set the requirement is they set an RDA or recommended dietary allowance. But when they don't have enough evidence to say how much we need for our health, they set an adequate intake or AI, which is basically a way of saying everyone else seems to be eating about this much and they seem to be okay, and so that must be adequate. So for pantothenic acid, we don't have an RDA. We have an AI because so little evidence was available in 1998 when it was created. But the idea that no one ever develops a natural pantothenic acid deficiency is so ingrained that it has essentially become a self-fulfilling prophecy that we don't have any evidence of natural pantothenic acid deficiency. In other words, because we believe that no one is ever naturally deficient in pantothenic acid, we don't really look for its deficiency anywhere. And because we never really look at its deficiency anywhere, we never find any evidence. And so there's a never-ending cycle of never finding any evidence because we don't look for any evidence, which we don't look for because there's no evidence that we would need to look. But is it true that no one ever naturally runs deficient in vitamin B5? I'll make the case to you today that it's quite possible that many of us need more vitamin B5, and that actually it's entirely possible that natural deficiency does exist and we just don't recognize it. So let's step back a little bit and talk about what these experiments looked like. So in 1976, there was a paper by Peggy Fry where she found that 9 weeks on a synthetic diet free of pantothenic acid compared to a control group that had the same synthetic diet but was supplemented with 10 milligrams per day of pantothenic acid, what they found was that in the deficient group, there was an apparent loss of energy and enthusiasm, and the people were complaining of fatigue. Now, one thing that people have pointed out is that this study didn't find the severe neurological effects that were produced by using an antagonist. But you could also point out that this study only lasted 9 weeks. What if those people had been on that deficient diet for a year or 2 years? My suspicion is that within a couple of months, extra, they would have seen much more severe effects. And if you imagine that someone is running a little bit low, maybe they're not on 0 pantothenic acid, but what if they're on 30% less than they need over the course of 5 to 10 years? What would happen then? We have no reason, based on the lack of an ability to produce severe deficiency in 9 weeks, we have no reason to believe that a moderate deficiency over many years wouldn't produce something severe. Now, before Peggy Fry had done that study in 1976, there were studies in the 1950s where they tried harder to induce pantothenic acid deficiency. And the way they did it was they put people on pantothenic acid deficient diets, and then they gave them an antagonist that would prevent the metabolic activation of the pantothenic acid into the forms of it that we need to use in our metabolism. And I'll talk later about the technical details of that. But for now, just think of it as a B5 antagonist. And so they reproduced, or I shouldn't say reproduced because this was done earlier, but they also produced fatigue, apathy, and malaise, which is general discomfort, uneasiness, or pain. Kind of feels like you're getting sick. They found that the subjects would start fighting with each other. They would appear sullen and childish. Sometimes they would spend the whole day in bed. As I said earlier in the story, they would all develop numbness and tingling in their hands and feet, and it will almost universally, they were wringing their hands or stamping their feet trying to restore normal sensation. They would have hyperactive reflexes. They would have weakness in the muscles of their hands and feet, and they would have an inability to walk on tiptoe. I just realized that I'm actually standing on my tiptoes right now as I'm speaking into the mic, so I'm happy that I don't have severe vitamin B5 deficiency. Now, some of them had a variety of unique manifestations of neurological problems. So in one person, they had the sensation of burning in their feet. In another person, they had a weak ability to bend the ankle so that the toes come toward the leg, and which is called dorsiflexion, and that led to an odd gait called steppage gait. This is where you're trying to walk, but because you can't bend through your toe normally, you have to pick the foot up off the ground a lot higher than you otherwise would have to. A third person had constant numbness in their hands. Some of the general effects outside of neurological problems were a pulse rate of about 120 in response to very minor exertion. Sleep disruption, insomnia was extremely common, and muscle cramps, especially abdominal cramps, and just gut problems in general, increased farting, nausea, occasionally vomiting. They also found that their eosinophils, which are white blood cells that are part of your immune system, were a lot lower than they should have been. And people, in fact, tended to get sick as a result of that. As I mentioned in the story, the symptoms started to recede after a few weeks of giving the antagonist, and they actually had to increase the dose of the antagonist. And so one of the things that we can see from this is that the body stores of pantothenic acid appear to be very large compared to what we eat on a daily basis. I'll put some numbers on that later. But basically, the reason that it was so hard to induce a deficiency in these controlled experiments is because it just takes a very long time to deplete the pool of pantothenic acid. And your body adapts to deficiency by trying to conserve it and trying to recycle it. But if you look at this, I mean, one of the things that you can notice is the symptoms of deficiency are not rare. Fatigue is not rare. Loss of enthusiasm is not rare. Arguing with people and being childish is not rare. Gut problems are not rare. Cramps are not rare. Insomnia is not rare. Feeling unfit is not rare. In fact, even tingling and numbness in the hands and feet isn't that rare. And of course, these things can have so many causes that you can't just go around saying your hands and feet are numb, therefore you must be deficient in pantothenic acid. But at the same time, if the symptoms aren't that rare, then maybe some of those symptoms some of the time are pantothenic acid deficiency. But because of the belief that pantothenic acid deficiency never occurs normally, what we have is all the studies where they were looking at the status of pantothenic acid in different populations, measuring blood levels and urine levels and looking at the correlation with dietary intake. They weren't doing questionnaires like, how often do you feel fatigued, or how enthusiastic are you about life, or even what is the incidence of abdominal cramping and farting, or any of the things that we would expect from the deficiency. So we have no idea from these studies if there's a level of dietary intake or blood or urine markers that correlate with the things that we would actually expect. So if we look at this, we could say, what are the signs and symptoms of maybe not clinical deficiency, but suboptimal status? And when we look at that, we see fatigue, loss of enthusiasm and energy, insomnia, grumpy mood, cramps, gut problems, these things are not that severe, and yet those are the major things that you find even in the deficiency studies. Based on some of the supplementation studies that Alex is going to cover, we could also posit that dyslipidemia, meaning high blood lipids, high cholesterol, high triglycerides, maybe are related to not having enough pantothenic acid, or at least getting extra might help with some of those things. Acne, and maybe poor wound healing. Now, on the flip side, is there pantothenic acid toxicity? Well, when the DRI was established in 1998 for pantothenic acid, they did not establish a tolerable upper intake limit, which is the level of supplementation or intake that you should avoid to provide a safety window against toxicity. They didn't set one because there was no evidence of toxicity. In fact, in 1952 and 1954, Welsh produced 2 reports of treating lupus with 10 to 15 grams per day of calcium pantothenate. 92%, I think, of that is actually pantothenic acid. So this is a tremendous amount. And he reported, quote, "It was found that transient nausea and gastric distress occurred about equally from the use of calcium pantothenate, which is one form that he used, sodium pantothenate, a second form he used, and pantotheniol alcohol, a third form that he used. He said symptoms of such distress occurred in only a few patients, and no patient was found who could not take one of the pantothenic acid derivatives studied. So very high doses with a rare incidence of GI distress from that. Alex, did you find anything about adverse effects of pantothenic acid supplementation in the studies you were looking at?
So I did, but nothing at doses that have been used in supplementation with purpose. For example, you mentioned the blood lipid effects. And when you look at the aggregate of all these studies through meta-analyses, the adverse effect rate is about 1.4%, where someone suffers an adverse effect. And it most commonly is going to be digestive distress, heartburn, and diarrhea. And so that's probably just from taking high doses moving through you. And these studies used anywhere from 600 to 1,200 milligrams per day. So nothing like the gram doses you used or were used in the study you mentioned. However, there was a really interesting study in children that used doses upwards of 1,000 milligrams per kilogram of body weight. And in these about 20 kids, the highest dose was 32 grams per day for about a week. And they were doing all of this to look at the pharmacokinetics. And they reported the exact same thing. The only side effect ever mentioned very infrequently and only at the highest dose was some GI distress and diarrhea, but no clinical signs of toxicity or any, really serious adverse events.
Cool. So to put this in perspective, the adequate intake, the AI, which is what we have because we don't have an RDA, is 5 milligrams a day for the typical adult. So when we're talking about 15 grams a day or 32 grams a day, we're talking about up to 6,400 times the adequate intake, with no evidence for anything other than some moderate GI distress in a small proportion of people. So it seems safe to say that there is no pantothenic acid toxicity, or at least if such a toxicity exists, it's at ridiculously higher doses than have been studied, which themselves are ridiculously higher than what most people would ever encounter. With that said, there is some hypothetical problems of taking high doses. And the main one would be that pantothenic acid and biotin share a transporter in the intestines. And so in theory, you could interfere with your biotin absorption with megadosing pantothenic acid. And of course, it's always possible that because many of the B vitamins are used together, when you megadose with one B vitamin, you might divert the others into certain pathways and wind up causing imbalances. But none of that has actually been shown definitively. And the incredibly high doses with no toxicity suggest that these are minor concerns. But I do think that it makes sense to shoot for balance in your diet and only use megadoses of pantothenic acid or another single B vitamin when you have a really good rationale to do so. So why is it the case that pantothenic acid deficiency results in the signs and symptoms that we were just talking about? Let's talk a little bit about what pantothenic acid is and what it does in the body. So first of all, what is it? Pantothenic acid is something that is synthesized by archaea, which are bacteria-like creatures, bacteria, fungi, and plants, but not by animals. And in all of these, what they do is they take pantoic acid, which is a short-chain fatty acid, and they take beta-alanine, which is an amino acid, and they combine them to make pantothenic acid. Life at all levels, including in those microorganisms and plants, but also in humans and other animals, we use 4-phosphopantothene as a cofactor for our enzymes, and we also convert that to coenzyme A. In 4-phosphopantothene, what we have is the sulfur amino acid cysteine, which you get from any dietary protein, but you will wind up with more of it if you eat animal protein. You take cysteine, you chop off a piece of it, and you get beta-mercaptoethylamine. And cysteine is a sulfur amino acid. It has sulfur. The piece you chopped off wasn't the sulfur, so beta-mercaptoethylamine has sulfur in it. And you stick that together to pantothenic acid. You stick it onto the beta-alanine end of that molecule. Then on the other end of the molecule, on the pantoic acid end of the molecule, you stick a phosphate there. You wind up with 4-phosphopantothene. This is one of the most important forms of pantothenic acid in the body. And then you can take 4-phosphopantothene and you can connect it to a slightly modified form of ADP. That's adenosine diphosphate, which is derived from ATP, the main energy currency of the cell. Both 4-phosphopantothene and coenzyme A, or CoA, are used as enzymatic cofactors that carry acyl groups. An acyl group is an acid that has the acidic portion chopped off so that you can stick it on something else. As an example, acetic acid is an acid. This is the acid that makes up vinegar. If you chop off the acidic portion, you wind up with an acetyl group. If you join that to coenzyme A, or CoA, you get acetyl-CoA. Acetyl-CoA is one example of many different acyl-CoAs. There are many other acyl groups that are carried by CoA, and 4-phosphopantothene carries these acyl groups as well. In fact, when we break down fatty acids for energy, we chop the fatty acid into 2-carbon acetyl units. So if we have a 16-carbon fatty acid called palmitate, we are going to join it to CoA to make a fatty acyl CoA. And the specific fatty acyl CoA would be palmitoyl CoA. And then when we start breaking it down for energy, we chop it off 2 carbons at a time. And each time we chop off 2 carbons, we join that to CoA and we get acetyl CoA. So we take this fatty acyl CoA and we break it down and break it down and break it down into a bunch of acetyl CoA. The difference between 4-phosphopantothene and CoA is that CoA takes things and moves them around. It transfers things from one place to another. It'll often be the thing that takes something from one biochemical pathway and puts it in another one. This makes it very much like niacin. If you were listening to the last 2 episodes in this series on niacin, and if you listen to the one before it on riboflavin, we talked about how niacin is often carrying energy around between different pathways and shuttling it around in different locations, whereas riboflavin is doing something very similar, but it's bound to the enzyme and participating in the transformation. Well, if you throw pantothenic acid into this mix, niacin and riboflavin are primarily carriers of the energy that you get when you break apart molecules. Coenzyme A and 4-phosphopantothene, these are carriers of the pieces of the molecules when you break them apart. But between the 2 forms of pantothenic acid, coenzyme A is very similar to niacin in the sense that it is taking these pieces of molecules and it's shuttling them around in different locations. Whereas 4-phosphopantothene is very similar to riboflavin in that it is bound to the enzymes as what we call in biochemistry a prosthetic group. That's kind of like if you have a prosthetic knee, it's part of you. A prosthetic group of an enzyme is part of the enzyme itself. Okay, so that's what the 2 major things that we do with pantothenic acid, 4-phosphopantothene and coenzyme A. That's what they are. Now, what do we do with them? Well, coenzyme A is used in the breakdown of fats, carbohydrates, and proteins to utilize the energy within them. It's used in the synthesis of fatty acids, ketones, and cholesterol. Because it's important to the synthesis of cholesterol, it is required for the synthesis of all steroid hormones, including all of the sex hormones, including the glucocorticoids that help regulate your blood sugar, and including the mineral corticoids that help regulate your electrolyte balance and your blood pressure. The addition of acetyl groups or fatty acyl groups to other molecules plays both regulatory and structural purposes. For example, the acetylation of histones increases genetic expression. Histones are proteins within your DNA, and your DNA is wound around them, like thread being wound around a bead. And you need to open that up to express your genes and make proteins from them. And acetylation with acetyl-CoA is how you unwind that DNA and open it up so that you can express your genes. Acetylation of peptide hormones can turn them on or off. As an example, alpha-melanocyte stimulating hormone is a neuropeptide that stimulates the production of melanin to pigment your hair, skin, and eyes, to suppress your appetite, and to increase your libido. Acetylation with acetyl-CoA turns it on. This might underlie one experiment where black rats were fed pantothenic acid deficient diets and they had gray hair. Perhaps the alpha-melanocyte stimulating hormone was not being acetylated properly. Beta endorphin is a natural painkiller and stress reducer. Acetylation turns it off. So acetylation with acetyl-CoA can turn one thing on and it can turn another thing off. Other acetylation reactions include the addition of the saturated fatty acids myristate, stearate, and palmitate to proteins. This serves to anchor them in cell membranes. It serves to tag them for specific locations in the cell. And it serves to create a reversible switch used to turn them on or off. As examples, adding palmitate to the transferrin receptor. This is the palmitoylation of the transferrin receptor. the transferrin receptor is critical to understanding your cells, understanding how much iron you have and what you should be doing with it. Palmitoylation is needed to regulate the transferrin receptor, and that's needed to utilize iron properly. In the nervous system, there are human conditions where within the nervous system, the palmitoylation of the transferrin receptor is defective, and that leads to neurodegenerative disorders where iron accumulates as ferritin within the brain cells. Ferritin is a storage protein for iron. And so in the brain cells, you have this deposition of iron, but it's all caught up in ferritin instead of being used properly for the things that it's supposed to do in the nervous system. The addition of stearate with stearoyl-CoA, this is the steroylation of proteins, is needed to hold mitochondria together. There are experiments in fruit flies showing that if you impair the steroylation of mitochondrial proteins, the mitochondria fall apart. They fragment into pieces. And there's evidence that this is involved in Parkinson's disease. Acetyl-CoA also modifies serotonin by converting it into N-Acetyl Serotonin. This is the precursor to melatonin. So what happens when you make melatonin, which is what makes you go to sleep at night, is that anytime during the day you get tryptophan into your brain, and that tryptophan is converted into serotonin. A portion of that serotonin, you use acetyl-CoA to acetylate it to N-Acetyl Serotonin, and this is then stored in your pineal gland, waiting to be converted into melatonin when it gets dark. You can store it all day long there. You don't have to get it in at night. Then at night, because of darkness, over the course of 2 to 4 hours of darkness, you start methylating that N-Acetyl Serotonin into melatonin. So you could have a methylation problem, and if you don't know what that is, head to chrismasterjohnphd.com/methylation. You could have a methylation problem interfering with your melatonin synthesis, but you could also have an acetylation problem because you don't have acetyl-CoA if you don't have enough pantothenic acid. Perhaps this explains the insomnia seen in pantothenic acid deficiency in the studies we talked about before. Acetyl-CoA also is used to acetylate choline to make acetylcholine. Acetylcholine is a neurotransmitter that promotes learning, memory, and sustained focused attention. Acetyl-CoA is also used to synthesize N-acetylglutamate. N-acetylglutamate is a regulator of the urea cycle. The urea cycle is how you get rid of ammonia that you get from protein. So every time you metabolize protein in any way, you generate ammonia. And ammonia is very toxic, so you need to get rid of it. You get rid of it by converting it to urea, which is named after the fact that it pops up in your urine. And urea is non-toxic. So what happens if you don't convert the ammonia to urea is that you smell funny and you feel like crap when you metabolize protein. And in fact, if the urea cycle is really messed up, then eating protein can kill you. That's extremely rare. But you could have hyperammonemia, the inability to clear the ammonia from their system, not just because you have a genetic defect in the urea cycle, but also because you don't have enough vitamin B5 in the system. In fact, there are other ways for you to run out of CoA. And so one of the things I'll talk about later is that there are many metabolic disorders where something is backed up that's hogging all the CoA and it stops you from making acetyl-CoA. And even though that disorder has nothing to do with protein metabolism, the drop in acetyl-CoA levels cause a drop in the urea cycle, and that causes hyperammonemia. The sugars that make up glycoproteins and glycolipids are all acetylated using acetyl-CoA. An example of this is mucin, which makes up mucus. This is the mucus that lubricates the eyes, the ears, the nose, the mouth, the vagina, the penis, and all the internal organs. Enzymes called N-acetyltransferases use acetyl-CoA to detoxify some drugs. CoA is used in the form of succinyl-CoA to combine with glycine using vitamin B6 in the first reaction that leads to the synthesis of heme. Heme is an important part of hemoglobin. Hemoglobin is how you carry oxygen to your tissues. Hemoglobin is why you are not constantly anemic. But heme is also used in other enzymes. For example, it's used in the cytochrome P450 enzymes involved in liver detoxification. This is two different ways that B5 is supporting detoxification. The cyclooxygenase enzyme, more commonly known as the COX enzyme, which converts the omega-6 fatty acid arachidonic acid into both things that cause inflammation, but also things that resolve inflammation and make it go away, and also things in the gut that prevent food intolerances. That enzyme is a cytochrome that uses heme. So you need B5 to make that enzyme. Catalase is an enzyme that uses heme involved in antioxidant defense. It helps protect your tissues from wear and tear as you age by getting rid of excess hydrogen peroxide. Because heme is part of that enzyme, vitamin B5 is needed to help you make that enzyme. In addition to all these roles of coenzyme A, there are also specific roles of 4-phosphopantothene. 4-phosphopantothene is a cofactor for acyl carrier protein, which is involved in fatty acid synthesis. In fact, acetyl-CoA is also involved in fatty acid synthesis. And so both coenzyme A and 4-phosphopantothene are needed to synthesize fatty acids. 4-phosphopantothene is also a cofactor for an enzyme that metabolizes folate. This enzyme is 10-formyl tetrahydrofolate dehydrogenase. That's a mouthful. Don't try to remember that. To put it simply, this is an enzyme that helps us make the unmethylated form of folate, THF, or tetrahydrofolate. And this is the form of folate that is used to make ATP, the major energy currency of our cell. And also half of the units that make up DNA and RNA are synthesized using the unmethylated form of folate. Also, the adenosine components of coenzyme A itself, of FAD made from riboflavin, and of NAD and NADP made from niacin are all synthesized in this pathway using the unmethylated form of folate. If you listened to our podcast on why you need glycine, we talked about how the unmethylated form of folate is the limiting factor for the synthesis of glycine. The reason that we fall short of how much glycine we need by an estimated 10 to 60 grams a day is because we don't make enough of this unmethylated form of folate. And it is 4-phosphopantothene, the derivative of vitamin B5, that allows us to make that unmethylated form of folate. 4-phosphopantothene is also a cofactor for an enzyme involved in the breakdown of the amino acid lysine, so it plays a minor role in protein metabolism as well. Now, when we look at these things, does it matter how much carbs and fat we eat? So we talked about how for thiamine, we need twice as much thiamine when we metabolize carbs as when we metabolize fat. And then when we talked about riboflavin, we talked about how we need 89% more riboflavin when we burn fat than when we burn carbs. So thiamine is the carb burner and riboflavin is the fat burner, and niacin is the thing that is just central to all of it. Is there any effect of our macronutrient ratios on how much vitamin B5 we need? Well, there is a minor effect that makes it more important for fat metabolism. So when we metabolize fat for energy, we require about 20% more coenzyme A from vitamin B5 than when we burn carbohydrate. So there's a modest effect of needing more B5 when we metabolize fat. With that said, there is some evidence that if you are deficient in B5, or at least if you're very deficient, or your metabolism of vitamin B5 is impaired, then a high-fat diet could be way more than 20% more stressful. So one thing to keep in mind is that even though completely oxidizing glucose for energy only versus completely oxidizing fat. Even though the difference is only 20% more with fat, it's also the case that when we are under stress, we can use anaerobic glycolysis to produce lactate from glucose, and that doesn't require any coenzyme A at all. That doesn't require any vitamin B5 at all. And that means that although normal metabolism only requires 20% more B5, it means that under stressed metabolism, you can go a long way without using any B5 at all if you burn carbs. But you cannot do that at all with fat. There is no way. There's no way whatsoever to burn fat for energy that doesn't require vitamin B5. And so an example of this is there is a genetic defect in the synthesis of coenzyme A from vitamin B5 that I'll talk about more later. And this defect causes neurodegeneration. So in trying to understand how we can fix this defect or what we can do about it, scientists have tried to make animal models of the defect. So they make mice that have that defect. And when those mice are given that defect, because the enzyme is distributed a little bit differently in them, in humans, we have a very brain-specific enzyme for making coenzyme A, and in mice, it's not so specific to the brain. So they do get some neurodegeneration when you make them defective in that enzyme, but it's not as powerful as it is in humans. But when those mice are put on a ketogenic diet, which is a high-fat, low-carbohydrate diet, all of a sudden the neurodegeneration becomes devastating. And so you look at this and you think, well, 20% more B5 for a high-fat diet, why would it make a difference? And it's because of this fact that when things are not operating optimally, when you are under stressed conditions, when you're at high intensity exercise, when you're in a race trying to beat the person next to you, when you're being suffocated and you need to find some way to survive long enough to get out of the suffocation, when your body's under any kind of stress like that, it starts diverting energy into lactate production. And it is able to harness a lot of energy from that. And it can do that without vitamin B5. So what are these poor mice on a ketogenic diet going to do when they have an enzymatic defect that prevents them from making coenzyme A? Suddenly, this ketogenic diet is robbing them of this ability to operate without coenzyme A under high stress. And I think that's what's driving them into the neurodegeneration. So if the system is running optimally, eating a high-fat diet is only going to slightly increase your needs for vitamin B5. But if your vitamin B5 system is not operating optimally, if there's something wrong with it, either because you're deficient dietarily or there's something wrong with your vitamin B5 metabolism, that can make you very vulnerable to getting wrecked by a high-fat diet. Now, as we move into talking about how we synthesize and degrade coenzyme A, it's important to give a broad perspective of how we would want to regulate the balance of CoA by thinking about the importance of CoA in regulating the many other things going on in the cell. So, first of all, the acetyl CoA to free CoA ratio is an important regulator of many metabolic pathways. It first is going to regulate its own production. So a high acetyl CoA to free CoA ratio will inhibit the production of acetyl CoA from glucose. And then it will say, you know what? We have enough acetyl CoA. So instead of making acetyl CoA from glucose, start using this glucose for other things, such as anabolic reactions, where you could synthesize other molecules. The acetyl CoA to free CoA ratio actually regulates many proteins by acetylation. And it also regulates many genes. And what happens is, although it's true in general that acetylating proteins can turn them on or turn them off, and although it's true in general that acetylating histones to unwind the DNA will increase genetic expression, it's actually more sophisticated than that, because the different histones of different genes or the different proteins that could be acetylated, they will get acetylated selectively at different acetyl-CoA to free-CoA ratios. In other words, you have a certain percentage of CoA is acetylated, and then you're flipping on one set of genes. But then you have more of your CoA acetylated. Suddenly you're flipping on a different set of genes. And so a high ratio of acetyl-CoA to free-CoA is going to increase the expression of genes involved in the fed state metabolism as opposed to the fasting state. For example, the genes to help you burn glucose for energy. It's going to increase the expression of genes involved in growth and replication. And it's going to increase the production of genes that invest energy in long-term protection, such as antioxidant defense. Conversely, a low ratio of acetyl-CoA to free-CoA is going to increase the expression of genes related to autophagy, which is breaking down things in your cell in response to stress. And depending on the level of stress, it may stimulate apoptosis, which is programmed cell death. If you think about how fat burning and carbohydrate burning play into this, fats primarily lead to high levels of acetyl-CoA in the mitochondria. Carbohydrates generate a lot of citrate in the mitochondria that then leaves the mitochondria into the cytosol, which is the general compartment of the cell. And in the cytosol, the citrate becomes acetyl-CoA. So carbohydrates increase cytosolic acetyl-CoA. That becomes the source of nuclear acetyl-CoA. So when you have high carbohydrate intake, you have a lot of nuclear acetyl-CoA regulating the genes. But when you're primarily burning fat, you have low levels of nuclear acetyl-CoA regulating the genes. The result of this is that fat burning, which is primarily a characteristic of the fasting state, you can eat a high-fat diet. But if you think of a mixed diet or a high carbohydrate diet, when you're burning fat is when you're fasting. And so fat burning, which is a signal of the fasting state, leads to lower acetylation of histones in the nucleus. And carbohydrate feeding leads to more acetylation of histones in the nucleus. The net effect is that fasting and fat burning promote autophagy and catabolism, while carbohydrate feeding promotes anabolism, building things up. Now, it's very important to realize that that people talk about different things in these pathways as having different benefits. It's important to realize why you need both. For example, many people talk about the benefits of autophagy. And autophagy is something you can do when you don't have a lot of energy, because by definition, it's breaking down stuff you already have, right? So autophagy is cleaning house. It's saying, I got a bunch of stuff I don't need. I can put this to better use. So maybe you get rid of damaged stuff. Maybe you get rid of stuff you don't need anymore, and you tidy things up and make them more efficient. But antioxidant defense, which is critical to protecting your tissues from damage long-term, is not something you can do in a low-energy state, because the entire antioxidant defense system runs on energy. And so antioxidant defense is only something you do in the well-fed state. So you can't go around saying fat is better because of autophagy. Carbohydrate is better because of antioxidant defense. Sometimes you need to clean house, but you need to spend most of the time in a high-energy state where you can actually defend your tissues from damage. And so it's natural to cycle between these states. Now, that also means, though, that if the ratios of acetyl-CoA to CoA get messed up, we're going to have problems. And so one of the things that happens is that the fact that the nucleus has histones that take in acetyl groups actually makes the nucleus a good buffer of the acetyl-CoA to CoA ratio. The histones in the nucleus can hold 1 billion acetyl groups in every cell. Think of a billion acetyl groups. You know how many acetyl groups you get from glucose? A molecule of glucose gives you 2 acetyl groups. So you have you have 500 million glucose molecules worth of acetyl groups that can be held in the nucleus. And so that means that under conditions of low energy supply, those acetyl groups can start leaving the nucleus and can keep the acetyl-CoA levels from bonking too low. While in a high energy state, the acetyl groups can go into the nucleus and prevent the acetyl-CoA levels from getting too high. And so that's good, but that buffer is not the only way we would want to control the acetyl-CoA to free-CoA ratio. So think of what the consequences of that buffer capacity being exceeded would be. If you have too much acetyl-CoA, you're going to have excessive growth and proliferation. If you have too much free-CoA, you're going to have excessive catabolism. You're going to have too much autophagy. You're also going to compromise your ability to dispose of ammonia. That's going to make you smell bad and feel like crap. You're also going to compromise your production of melatonin. That's going to interfere with your sleep. You're also going to compromise your production of acetylcholine. That's going to interfere with your focused concentration. You're also going to interfere with your mucin. That's going to make your eyes, nose, mouth, penis, vagina all dry. And we are going to want to keep these ratios balanced. In addition to this, as I mentioned before, there are a number of metabolic disorders that lead to hogging up the CoA pool. When they do so, they lead to hogging CoA up in an acyl-CoA that reflects the metabolic disorder. For example, you may have a fatty acid oxidation disorder. These are all rare, but there are so many of them that collectively that you're bound to run into someone that knows someone who has one of these things at some point. If you have a fatty acid oxidation disorder, then maybe you can't burn medium chain fatty acids. So you take a long chain fatty acid, you start chopping it off into acetyl groups, and then you get down to a medium chain fatty acid. Suddenly you can't burn it anymore. So now what do you have? You have a medium chain acyl-CoA. So at first, like let's say you drank a cup of bulletproof coffee, and well, actually, let's be generous and say you put butter in the coffee instead of MCT oil. The MCT oil would kill you very quickly. But let's say it was butter and you got some long chain fats. You start chopping off acetyl groups. At first, you're all good. You got plenty of acetyl-CoA. Things are operating nicely. But then you chop that down to a medium chain acyl-CoA. You got nothing to do with it. You can't burn it anywhere. You just got a medium chain acyl-CoA, and you're like, what do I do with this thing? Well, I'm going to put it over here for now, and I'm going to burn the next long chain fatty acid in that butter. Chop off some acetyl groups. You still feel good. Now you got another medium chain acyl-CoA. Well, you keep doing that, and for a while, you're not going to chop off any more acetyl groups to make acetyl-CoA because all your CoA is stuck in these medium chain fatty acyl-CoAs that you can't do anything with. And so this then mimics having too much free CoA. What happens? You have excessive catabolism. You compromise your ammonia disposal. You smell funny. You feel like crap. You can't sleep because of melatonin. You can't focus because of acetylcholine. You are dry all over because of mucin. Okay, so now when we look at the synthesis and degradation of coenzyme A, we want to keep in mind that there's got to be ways to prevent the free CoA pool and the total CoA pool from going out of whack. So how do we do this? Well, if we start from pantothenic acid, which we remember, this is a form that plants can make it, bacteria can make it, fungi can make it. It is a minor portion of what's in our food, and it is a big portion of what's in our supplements. But let's start there for simplicity. For pantothenic acid, first we are going to add a phosphate to it to make 4-phosphopentothenic acid. Then we are going to take cysteine and stick it on because this is going to be the source of our sulfur. So we stick on cysteine and we make phosphopantothenoyl cysteine. Then we break that off a little bit to modify it to the form that we want. And we get phosphopantothene, or 4-phosphopantothene. 4-phosphopantothene has the sulfur that the cysteine had added. Then we add ADP to it, and we get D-phospho-CoA. Then we add another phosphate to it, and we get coenzyme A, or CoA. So when we do this, the first step, which is taking place in the cytosol, the main compartment of the cell most of the time, or at least it's thought to mostly occur there. We convert pantothenic acid to 4-phosphopantothenic acid with an enzyme called pantothenic acid kinase, or PANK. And this enzyme requires magnesium and ATP. Recall that when we talked about riboflavin and we talked about niacin, we talked about them getting activated using magnesium and ATP. And we said how having food in our system, so we have lots of ATP, and having magnesium is needed to metabolically activate them. That's also true for pantothenic acid. In step 2, we take phosphopantothenic acid and we add cysteine to it. And that leads to phosphopantothenoyl cysteine. That requires magnesium. It requires ATP, just like the first step. And it also requires cysteine. Where do we get our cysteine from? Well, we need to eat enough sulfur amino acids. This comes from any of the protein in our diet, but there's twice as many in animal products as plant products. And then we need to use vitamin B6 and glycine in order to make that cysteine. So when we're going up this pathway, we don't just need magnesium and ATP, we also need vitamin B6 and glycine. Then in step 3, we take phosphopantothenoyl cysteine and we make 4-phosphopantothene. Once we make the 4-phosphopantothene in this step, we can then use it as a cofactor for the enzymes that use 4-phosphopantothene that I had described before. But if we need coenzyme A, we go on to step 4. This is where we add ADP to it to make D-phospho-CoA. This step uses magnesium and ATP. Then step 5 is we take D-phospho-CoA and we add phosphate to it. This step uses magnesium and ATP. These last 2 steps occur in the mitochondrion, at least most of the time it's thought, and they occur with a multi-enzyme complex where it's actually a factory made of these 2 enzymes, and that factory is called coenzyme A synthetase. So we regulate this pathway to make sure that our CoA levels don't go out of whack. In the liver, the key regulated step in any tissue is pantothenic kinase, the first step. In the liver, pantothenic kinase is inhibited very strongly by 4-phosphopantothene and by free coenzyme A. So the 2 end products that we need tell the enzyme that we have enough and they shut down their own production. To a lesser extent, acylated CoAs are going to inhibit the pantothenic kinase enzyme. In other words, you inhibit it very strongly if you have too much free CoA, and you inhibit it a little bit more if you have too much total CoA. In heart, the inhibition is a lot stronger, and the total CoA pool is almost as strong as the free CoA pool. So in heart, the heart is just sort of itching to have an excuse to stop the pantothenic kinase enzyme. If these were the only regulators, the activity in heart would almost always be 0. Carnitine, which helps transport fatty acids into the mitochondrion to be burned for energy, which increases your need for coenzyme A, relieves that inhibition. But still, at maximal carnitine concentrations, that would only bring the enzyme up to 13%. So there are other regulators that we don't really understand that much in heart. And heart appears to normally operate far below its normal capacity under most conditions. And in fact, if you look at the tissue concentrations, the heart will usually have a much larger percentage of its pantothenate as free pantothenate that can be made into coenzyme A, but isn't. And so the heart has a large reserve capacity to make more CoA when it needs to, whereas the liver just has way more CoA, and it usually doesn't have a lot of pantothenate around. It usually just has tons of coenzyme A. And so you can, if you think about that, think of the heart is primed to be able to make more CoA when it needs to, and when it needs to is really under high physical activity, right? Your heart starts pumping a lot more if you start running or if you start lifting weights. And so the heart needs to go back and forth in terms of how much coenzyme A it's using to burn fatty acids. The liver, meanwhile, the liver is the metabolic hub of the body. It's always doing something. And so it always needs to have a lot of coenzyme A. Also, If the liver has a huge coenzyme A pool, then that is going to make it very affected by regulation. So because there's so much free coenzyme A, it's very sensitive to eating carbohydrates, leading to greater expression of the fed state genes through that acetyl-CoA to CoA ratio. Whereas carbohydrate intake drops, and now that ratio changes a lot, and now the liver will shift to fasting state physiology. And so the liver having this huge CoA pool so that the ratio will change a lot in response to fat feeding versus carbohydrate feeding is a way to allow the liver to engage in very sensitive responses to cues from the environment about what it should be doing for the rest of the body. There are some other regulatory factors that we know play in here. So under fasting conditions and in diabetes, the liver and heart CoA go up. This is because insulin suppresses coenzyme A synthesis in both tissues. And that makes sense because when your insulin levels drop, you have increased demand for coenzyme A because you're burning more fat. Glucocorticoids, thyroid, and cold exposure all increase the synthesis of coenzyme A in the liver. These are all things that increase the movement of fatty acids and amino acids to the liver for gluconeogenesis. So the liver will have much higher demands under those conditions to burn fatty acids for energy and to use acetyl-CoA in synthetic reactions. Now, as I noted before, it's not just coenzyme A that we use. We also have to add 4-phosphopantothene to enzymes that use it. And the main way that we do that is we take coenzyme A and we have a special enzyme that will break apart the coenzyme A to add to the proteins that use 4-phosphopantothene. Now let's shift to talking about the degradation of coenzyme A. And this is a pretty interesting topic, but it's fairly poorly understood. So we know that coenzyme A can have its phosphate chopped off in specialized digestive components of the cell called lysosomes. But this appears to be very strongly inhibited by phosphate and ATP and seems to be something that you would only do when you're looking for phosphate that you need to find from somewhere. You'll break some coenzyme A down to get it. It's probably the case that the first step doesn't operate, the one I just mentioned, doesn't operate that often. The second step is we can take D-phospho-CoA that we got from the first step, or we can do this with free coenzyme A or even acyl CoAs. We can break them down to 4-phosphopantothene. And this is using an enzyme that is not specific to this process, but it just generally can break down nucleotides. So it can also break down ATP, for example. Now, at this point, we would have 4-phosphopantothene. We could recycle it and salvage it, or we could continue breaking it down. If we continue breaking it down, we go on to step 3, and we cleave off the phosphate to make pantothene. That pantothene, now we can salvage that, we can recycle it, or we can continue breaking it down with an enzyme pantothenase. And in fact, this enzyme pantothenase, this is the first time that pantothene-specific enzymes appear to be involved in this process. And that pantothenase will break the pantothene down to pantothenic acid and another product called cysteamine. And that pantothenic acid, again, we can recycle it, we can salvage it, or that is what we pee out in the urine. Then the cysteamine that's left over can be converted into taurine, which plays important roles in the body. If you look at this degradation pathway, it first of all doesn't happen in the mitochondria. And in the mitochondria in liver, some 50 to 80% of the coenzyme A is in the mitochondria. In heart, 95% of the coenzyme A is in the mitochondria. So the first step is actually for the CoA to leave the mitochondria if mitochondrial CoA is going to be degraded. So it may wind up being that the cytosolic CoA pool is degraded at maybe a fairly constant rate in proportion to the amount of CoA there, just to keep the CoA pool in the cytosol, which remember is the one that's influencing nuclear gene expression, within fairly confined limits. But if the majority of the CoA, and maybe even the overwhelming majority of CoA is in the mitochondria, then the degree to which you degrade CoA might be it might be most strongly influenced by the mitochondria spitting out their CoA into the cytosol where it can be degraded. There's a tremendous amount that is not understood about how degradation is regulated. It's very clear that synthesis is strongly regulated by your need for phosphopantothene and your need for coenzyme A. It's probably the case that degradation is also regulated by your needs for those, but also by your need to control the acetyl CoA to CoA ratio and the total CoA pool to make sure that your gene expression is not being distorted, to make sure that your regulation of proteins is not going out of whack. Okay, so let's shift gears now. We've talked about the biochemistry. Let's talk about the physiology. How do we extract this from food and transport it into our body and take advantage of it? So in humans, about 50% of the food in pantothenate is absorbed. Food pantothenate is mostly coenzyme A and 4-phosphopantothene. The proportions are unknown for most foods, but it's probably about 15% free pantothenic acid and 85% coenzyme A and 4-phosphopantothene. Although these can be completely digested down into pantothenic acid, because the digestion seems to be slow and incomplete, as is made clear by the fact that we only absorb half of it from food, that slow rate of digestion probably allows for the absorption of pantothene and 4-phosphopantothene. And I will make an argument later that food B5 is superior because we can more efficiently make our enzymatic cofactors from those forms. Now, in a 1983 study that is cited in every major review and is one of the most authoritative studies on how we digest coenzyme A, they showed that alkaline phosphatase and pantothenase are two enzymes present in the intestines, and they break down coenzyme A. They first take away the phosphate to make D-phospho-CoA. Then they take away the AMP to make 4-phosphopantothene. Then they take away another phosphate to make pantothene. And then alkaline phosphatase at this point cannot engage in further digestion, but pantothenase can further break down pantothene into pantothenic acid. In the DRI report in 1998, of the Food and Nutrition Board of the Institute of Medicine that set the adequate intake for us, they reviewed this paper in 1983, and they quote, this is what they said about it, quote, "Pantothenic acid was the only one of these pantothenate-containing compounds absorbed by rats in studies on absorption of the various forms." And the implication there is pantothenic acid in supplements is just as good as coenzyme A and 4-phosphopantothene in foods. But this statement is manifestly false. Let me read it one more time. Pantothenic acid was the only one of these pantothenate-containing compounds absorbed by rats in studies on absorption of the various forms. And they cite this paper that I'm reading to you now, published by Shibata and colleagues in 1983. Let's look at the data from that paper. After 30 minutes of coenzyme A, and this is not food, right? So food would be digested less efficiently and more slowly. They fed purified coenzyme A. And the coenzyme A, after 30 minutes in the small intestine, the major products were 40% of it as 4-phosphopantothene, 40% of it as pantothene, and 20% of it as pantothenic acid. So it's 20% as free pantothenic acid, 80% as the higher derivatives. When they looked at the passage of these compounds into the enterocyte, the intestinal cell responsible for absorbing our food nutrients, at 15 minutes, 36% of what was absorbed was pantothenic acid, 38% was pantothene, 28% was phosphopantothene, or things even higher than that, such as coenzyme A. At 1 hour, 47% was pantothenic acid, 24% was pantothene, 35% was phosphopantothene or higher. At 3 hours, 69% was pantothenic acid, 5% was pantothene, and 46% was phosphopantothene or higher. Over those 3 hours, there was a constant movement of the B5 and its derivatives to other tissues. That movement had already gotten going by the 15-minute mark. And by 3 hours, it was 40% complete, and it was nearly 100% complete by 5 hours. So when you're looking at those percentages, you have to imagine that the B5 is constantly leaving the enterocyte, right? So the fact that at 3 hours, we went from between 15 minutes and 3 hours, we went from 38% pantothene to 5% pantothene. That doesn't mean the pantothene was all being broken down into the enterocyte. It's perfectly consistent with all the pantothene leaving the enterocyte and going to other tissues. Same thing with the phosphopantothene and above. When we're looking at 28% at 15 minutes, it's actually going higher at 1 hour, and at 3 hours, it doesn't mean it's all accumulating there. There's a constant dynamic where it's being absorbed and it's passing to the other tissues. Now, they did take the intestines out of the live animal, and when they took out the intestines out of the animal and looked at absorption, it was 90% free pantothenic acid, only 10% pantothene, and 0% higher derivatives. But they concluded from that it was so remarkably different from the live animal that it obviously wasn't a good model. Now, they noted in their own study that they showed that you could absorb pantothene, 4-phosphopantothene. And they even commented on how it would make a lot of evolutionary sense if you could do that, because what you would want to do is conserve the enzymes and the ATP that you would need to convert pantothenic acid into coenzyme A. Because remember, we have 5 steps, almost all of which require magnesium and ATP, and some of which require other things as well, right? So it's hard to turn pantothenic acid into coenzyme A. You would want to save your resources. But then they cite a paper published by Ono and colleagues in 1974 as showing that even though you can absorb all these different forms into the enterocyte, only pantothenic acid leaves the enterocyte. Then they write, 2 enzymes and extra ATP are needed to synthesize coenzyme A when starting with pantothenic acid. Yet this non-conserving sequence seems to be the one used by the rat. Let me translate that into a little bit clearer English. They're saying, you would think that if we showed in our own paper that you absorb 4-phosphopantothene and pantothene, you would think you would want to save the ATP and save the enzymes by making more efficient synthesis of the cofactors that you need for your metabolism. But instead, we look at this other paper by Ono from last decade, and based on their paper, then what we think is going on is that the rat just wastes tons of energy by breaking down what it's absorbed into free pantothenic acid and then building it back up. Okay, so let's go look at the Ono paper from 1974, the one that they're using to contradict their own logical rationale of what the rat should do based on their own data. Ono and colleagues fed rats pantothine, first of all. They didn't feed coenzyme A and they didn't feed pantothene. Pantothene is spelled E-I-N-E. Pantothine is spelled I-N-E. There's no E in it at the end. And pantothine is 2 molecules of pantothene that have been oxidized and stuck together. Pantothine breaks down into 2 molecules of pantothene in the intestines. And in this study, the rats had 75 to 80% free pantothenic acid showing up in their blood. And so 20 to 25% of the oral dose of pantothine was not free pantothenic acid. And they provided no data suggesting that only free pantothenic acid leaves the enterocyte. In fact, if only free pantothenic acid leaves the enterocyte, why is 20 to 25% showing up in the blood as higher derivatives? Also, since metabolites are moved very rapidly from blood to other tissues, especially muscle, as we'll see soon, we can't rule out that the flux, right, the concentration of the higher derivatives was 20 to 25%. We cannot rule out that the flux through the blood to the muscle was far greater than 20 to 25%. And so when we come back to the Shibata paper in 1973, we can say that Shibata and colleagues were probably right that when they showed in their own paper that we absorb pantothene and 4-phosphopantothene, that we do use it more efficiently. But then you come back to the 1998 DRI report. I'll read this quote again. Shibata in 1983 showed, quote, pantothenic acid was the only one of these pantothenic-containing compounds absorbed by rats in studies on absorption of the various forms. This is just manifestly false. So we can conclude instead that we don't know a lot about this, but we do know that we absorb pantothenic acid, pantothene, and 4-phosphopantothene, and that we do wind up with the higher derivatives in our blood. And so some unknown proportion that might be very large is these higher derivatives absorbed intact that go to our cells and are more efficiently made into coenzyme A. More recently, in 2015 Nature Chemical Biology paper, Srinivasan showed that 4-phosphopantothene crosses membranes easily in certain types. This was remarkable to me because even cell biology textbooks say that things with charged phosphates on them don't pass through cell membranes because the charged phosphates makes them stick to the water. So I emailed the head of the laboratory that published this paper, and I said, I thought that charged phosphates can't cross membranes passively because of their high rate of hydrogen bonding with water. What about the 4-phosphopantothene molecule allows it to pass through the membrane so easily? He wrote back to me, you are asking an excellent question and we do not know the answer. We did demonstrate that in insect cells, obviously, it is possible for 4-phosphopantothene to somehow pass through membranes. So we have to reformulate our assumptions regarding this molecule. We hope to answer this intriguing question in the near future. So it's quite possible that the 4-phosphopantothene is just crossing easily through the membranes, and that would allow it to start absorbing even earlier than the small intestine. So when you eat food, you have coenzyme A and 4-phosphopantothene as the main forms of vitamin B5. And probably the 4-phosphopantothene is stuck to protein, so it's not going to start absorbing in the mouth. But then when it gets to the stomach and the proteins start getting digested and you free the 4-phosphopantothene, if it does slip through human stomach membranes like it slips through the insect membranes in their experiment, then we might just start absorbing the 4-phosphopantothene in the stomach before it ever comes in contact with the cell enzymes in the intestines that would break it down further. Almost everything that we know about pantothenic acid absorption is based on the idea that pantothenic acid is the only thing that's absorbed. No one's even tried to characterize how we would absorb other things because we're all operating on the assumption that pantothenic acid is the one thing that's absorbed. And what we know about pantothenic acid is that it is absorbed using the energy from sodium. And so usually you provide that sodium gradient using energy, but if your energy status is tanked, you might benefit from adding sodium to your food to absorb vitamin B5. And unlike niacin and riboflavin, you do not need to invest ATP energy and magnesium to keep it locked in the cell. And so in this case, it probably doesn't matter if you consume it with food. You might have better immediate synthesis of coenzyme A and 4-phosphopantothene if you consume it with food because of the higher energy status, but that's not going to be the thing that prevents you from losing it in the urine like it would be for niacin and riboflavin. So we absorb it from the intestine, and it's going to go into the portal vein into the liver. The liver likewise is going to take it up using sodium and not have the need to metabolically trap it using magnesium or ATP. And then the liver is going to distribute it to the rest of the body through the general circulation. So what does transport in the blood of pantothenate look like? Human pantothenic acid is carried about 90% in red blood cells, erythrocytes. And it's about 3 to 8% free pantothenic acid. So over 92 to 97% of the pantothenate in blood is higher derivatives such as 4-phosphopantothenic and coenzyme A. What we have only known for the last few years from the Srinivasan paper is that it may be the case, contrary to everything that we thought before, that the major circulating form of vitamin B5 is 4-phosphopantothene. Because what happens is cells can release coenzyme A, and the coenzyme in the A in the blood can be digested by enzymes to 4-phosphopantothene. 4-phosphopantothene is stable in the blood. It can spend hours in the blood, and it also may be easily taken up by cells by passively going through the membranes. If so, it's quite possible that 4-phosphopantothene is the major circulating form of vitamin B5. Now, it doesn't hang around in the blood. In 1969, Baker and colleagues injected humans with 45 milligrams of pantothenic acid. 4% went to the blood, 42% went to the urine, and 54% was taken up by tissues that were not blood. In the first 3 minutes, the blood concentrations were highest, so this transfer was rapid. And over the course of 24 hours, the pantothenic acid moved from plasma into red blood cells. Again, the small amount of pantothenic acid that was in the blood moved from plasma into the red blood cells. In 1976, Peggy Fry and colleagues found that 100 milligrams of pantothenic acid per day for 1 week was 60% retained. That corresponds remarkably to what Baker found in 1969 with 45 milligram injection. Baker found 60% retention, 42% into the urine. So almost 60% retention. Peggy Fry found 40% goes to the urine, 60% retained. So very different doses, 45 versus 100 milligrams. Very different model of delivery, injection versus eating. Very similar retention. But what's even more remarkable about this is the adequate intake for pantothenic acid is 5 milligrams. 100 milligrams is 20 times that. 45 milligrams is 9 times that. And you are still retaining 60% of it. So your ability to mop up huge doses of pantothenic acid is very strong. Now, in terms of how it gets from the blood into other cells, everything we know is based on the assumption that it's just pantothenic acid going into the cells, and yet we actually have known since the 1970s that free pantothenic acid is only 3 to 8% of what's in blood. And so those cells take up, in the brain, it gets in by passive diffusion. In the kidney, it's taken up by metabolic trapping, kind of like niacin and riboflavin. You needed to invest ATP to lock it up. That's how you get it into the kidney. That's how you keep it in the kidney. In adipocytes, it's taken up using sodium. And in certain cells within the brain, even though it gets into the brain passively, certain cells in the brain use energy and sodium to take it up. And the heart takes it up with sodium. But again, Srinivasan showed in 2015 that 4-phosphopantothene may be the main circulating form and may be taken up passively through diffusion across membranes. Now, what do we know about the tissue distribution? Well, I don't know how much pantothenic acid is in the body, and there may be someone in the world who does, but in my reading all the papers of the people that I think would know, it doesn't seem that there's any measurements of it. So what I did was I looked at the concentrations in rat tissues, and I didn't have all the rat tissues. I just had a bunch of the ones that were measured. And I looked up human organ weights, and I made a guesstimate, assuming that humans have similar concentrations as rats. And so these numbers are not to be trusted in their details, but this number will just give you a general sense within an order of magnitude of how much there might be. So I estimate that a human has about 4.5 grams of pantothenic acid stored in the body. And this isn't probably an underestimate, because none of these studies measured for phosphopantothene. They just measured free pantothenate and coenzyme A. So we probably have 4.5 grams. We probably have more than that. In rats, the highest concentrations are in liver, kidney, and heart, but all organs have some that have been measured. And skeletal muscle, even though it has a fairly low concentration, has the highest total amount simply because there's more skeletal muscle than there is of any other metabolically active tissue. When you dose rats with labeled pantothenic acid, you can watch what tissues it goes to. And very quickly, over the course of 5 hours, the pantothenate goes 35% to the muscle, 12% to the liver, 5% to the intestinal tissue, and 5% to the kidneys, and very little winds up in blood. Only 2% goes to the heart. And that's quite different from if you just kill a rat and you look at the total amount of pantothenic acid in those tissues. It's very different from what you see when you immediately eject it in, because again, the highest concentrations are in liver, kidney, and heart. And when you give them a dose, that acute dose is going more to the muscle. It's not going very much to the heart, and it's only going a little bit to the kidneys. When you excrete pantothenic acid, it's mostly in the urine, and it's mostly as free pantothenic acid. I talked before of how it was degraded. Well, all of those steps in the degradation pathway, you can salvage the pantothenic acid, but whatever you do not salvage gets spilled into the urine. In adolescence, there may be a particularly high need for pantothenic acid. There was an experiment where they fed weanling rats, so very young rats, with a very high rate of growth, a pantothenic acid deficient diet. Their hair turned gray. As they got older, their hair turned black. When they were younger, their urinary pantothenic acid was very low. But as they got older, it kept increasing, all on the same diet, suggesting that their needs for pantothenic acid when they were growing were dramatically higher than their needs when they were adults. And one paper that I read suggested, quote, adolescence may represent a period of peculiarly high pantothenic acid need. Later, Alex is going to talk about acne, and one of the authors of the paper on acne suggested that the need for vitamin B5 in adolescence would be very high because of the role of acetyl-CoA in generating cholesterol and sex hormones. In pregnancy, we know that in the 3rd trimester and 2 weeks to 3 months postpartum, the blood concentrations of pantothenic acid decline. The urine concentrations do not. And I'll talk later about why I believe that urine concentrations are a fairly faithful reflection of whole body stores. So what this indicates is that mothers are actively transferring pantothenic acid to their fetuses at their own expense and into their milk at their own expense, but it's not taxing their whole body stores all that much. It's primarily just tanking their blood levels. If you look at umbilical cord blood when a baby is born, it's 40% higher than maternal blood in pantothenic acid and all of its derivatives. And if you look at the mother, her blood is 44% lower than what you find in someone who's not pregnant. So basically the mother that's evidence the mother is actively nourishing the fetus at her own expense. In a study on postpartum girls, they had higher intakes of pantothenic acid because they were all taking their vitamins, their prenatals. And because of that, compared to girls who were not pregnant, they had higher urine pantothenic acid, reflecting higher whole body stores. But their blood was still lower. And so they're eating more. They have good body stores. They're urinating a lot out of it, but they are actively using all of that to nourish their fetus to the point that their blood levels drop. In lactation, milk pantothenate reflects your diet and not your blood. So supplementation with pantothenic acid to raise the pantothenate content of your diet 5.5-fold will increase your milk pantothenate by 87%, so almost doubling it. It has much less effect on the blood. And again, that's reflecting the active transport of pantothenic to nourish the fetus and then the nursing infant. So this is what I get from studies that were just looking at basic metabolism in this way. I know, Alex, you looked at some supplementation studies. Can you shed any additional light on the pharmacokinetics, how these things are transported and distributed based on what you looked at?
Okay, so we do have some data using pantothine, which again is the form of pantothenic acid where pantothene is bonded together by a disulfide bond. So it's basically two molecules held together as one. And in that child study I mentioned earlier, at the very highest tested dose of about 32 grams per day, which was 1,000 milligrams per kilogram, we found that almost 2/3 of the dose was not broken down or absorbed. It was completely unchanged in the feces of the children. And another 12 to 14% was broken down into pantothenic acid, but it wasn't absorbed. It was also present in the feces. And comparing this to lower doses and using some regression analyses, the researchers suggested that pantothine doses higher than 250 milligrams per kilogram, which corresponded to about 8 grams, was an upper limit for absorption, because after that amount, there was little further increase in serum levels of pantothenic acid or cysteamine, which is the name of the molecule that is going to hold the pantothene molecules together within pantothine. And so by measuring serum levels of that, it gives an indirect indication of how much pantothine made it across the intestinal wall, since it's going to be cleaved off from the molecules.
Did they, in those studies, did they look at, did they give any estimate of the proportion of what was retained by the body versus urinated after it was absorbed?
Kind of. They found that there was really two distinct pharmacokinetic responses. One is a linear, where as the dose gradually increased, the amount being retained gradually increased. And the other was an exponential one, where there was a very, very rapid and sudden increase in pantothenic acid stores relative to the excretion, that then quickly plateaued and leveled off. And the only difference between the children following one curve versus the other was their baseline levels of pantothenic acid in the body. And so the researchers hypothesized that if you start with lower levels, then your body's going to rapidly uptake and hold on to whatever you supplement. Whereas if you start with higher levels, that effect isn't going to be seen. Instead, it'll be more linear.
That's actually really fascinating. And I think I want to come back to that study later when we talk about how much we need, because I have some, it makes a lot of sense to me that you'd be mopping up huge amounts there. Did they quantify at all, like so much, like if 62% went into the feces and the rest was absorbed, did they do a 24-hour urine to see what proportion went into the urine? Or were they just using markers in the blood to try to like estimate the relative retention by the two different types of children that they divided?
Yeah. So the researchers, what they did is they did random stool samples, but then they also collected urine samples over a 3-day period with the doses. And so to reiterate, what they found was 62% of the pantothine dose was absorbed or broken down in the intestinal cells and was just excreted intact. And then another 24% was broken down but still excreted. It wasn't absorbed, which puts us at about 86% of the total dose being lost in the feces. So of that 14% that was absorbed, after 3 days, there was an 8% excretion in the urine. And so overall, the body retained about 8% of the highest pantothine dose, which was 1,000 milligrams per kilogram.
Okay, so if we take that number of at 1,000 milligrams per kilogram, you're retaining 8% of it. If we just sort of assume that we can transfer this from children to adults, we would get the average reference 70 kilogram man, 150-pound man would retain 5.6 grams of that dose.
Yeah, but we also need to keep in mind that, children and most, 30 to 40% of pantothenic acid is stored in muscle tissue. So as you age, you're going to put on more muscle tissue. And then if you also look at, physically active, like resistance training people who then put on even more muscle tissue, it's logical to speculate that simply the more muscle mass you carry, the more storage you're going to have of pantothenic acid.
Yeah, and we also have to keep in mind that we know almost nothing about how the different developmental stages are changing the turnover rate. So we speculated from that one experiment in rats earlier that adolescence, because of its rapid growth, is increasing your needs. But this is sort of symptomatic of the widespread belief that it doesn't matter. And so there's just so much that we don't know about how the needs change over time. But in any case, this indicates that even though Alex is talking about a pantothine supplement, which is different than what I was talking about when we had pantothenic acid or pantothenate, which are identical terms, being orally dosed at 100 milligrams or injected at 45 milligrams. What's remarkable about all these studies is even if we don't trust the exact details of the numbers, we are talking about the ability to retain huge amounts of megadoses of this stuff. This is so radically different from when we talked about riboflavin, for example, where you take 10 milligrams of riboflavin and your pee is neon yellow from the riboflavin that you're peeing out. And you are able to, when we talk about riboflavin, you were able to retain whatever you could activate into FAD at that time, and then the rest is whoosh, right in the urine. Pantothenic acid seems to have these very deep body stores where you will mop it up like a sponge and hold on to it.
So to give you an idea of how much this amounts to as a vitamin being stored in the body, the average well-nourished person in industrialized countries store 4 to 5 grams of iron in their bodies. So you're storing as much pantothenic acid in your body as you are iron.
Yeah, although the calcium and phosphorus would probably be even bigger, right? But yeah, so these are total body stores of a nutrient can be on gram levels or smaller than that or bigger than that. And it looks like pantothenic acid total body stores are on the order of grams. So whatever we can't say about it, we have grams of it in our body. And we can build those stores up so fast is what's crazy here. All right, I want to end part 1 by taking what we learned about the biochemistry and the physiology and making a case for why the best form of vitamin B5 is from food. And notice that when we covered riboflavin, I went into it thinking riboflavin 5-phosphate was superior to free riboflavin and came out of it saying it doesn't matter and free riboflavin might be best. I've come out of pantothenic acid research looking at the major reviews, for example, the DRI report saying that you don't absorb anything except pantothenic acid. And I've come away from it thinking that you can make an extremely strong case that for vitamin B5, food is superior. that it is superior to get coenzyme A and 4-phosphopantothene, the major forms in food, compared to taking pantothenic acid supplements. And at the end of this, I will make the case that pantothine, which is what they were using in the study Alex was talking about, is better than pantothenic acid or pantothenate. But I really believe that food is superior. So let's go through the case here. Food contains 85%-ish coenzyme A and 4-phosphopantothene in probably variable proportions across foods, but generally in any food, the proportion is unknown. Coenzyme A is digested into pantothenic acid, 4-phosphopantothene, and pantothene in the gut, like we talked about before. The pantothene can generate 4-phosphopantothene and coenzyme A more easily in the intestinal cells by skipping the second and third steps of synthesis on the way to 4-phosphopantothene, one of which requires ATP. Now, the 4-phosphopantothene, which is part of what you digest CoA into, and which is also itself found in food, may be absorbed intact. The 4-phosphate group on 4-phosphopantothene makes it resistant to the pantothenase enzymes compared to other B5 derivatives. And based on the 2015 paper by Srinivasan, the 4-phosphopantothene is passively absorbed through membranes. So we might start absorbing it in our stomach when we start digesting the food proteins that it's bound to, in which case we might absorb a lot of that 4-phosphopantothene intact. Then in the intestine, when we start breaking down the coenzyme A, we might absorb a lot of the 4-phosphopantothene intact. And then we also absorb some of the pantothene intact. When you absorb 4-phosphopantothene, you have skipped the first 3 steps of synthesis, and you only have to go through the final 2 steps in the mitochondrion. You do need magnesium, you do need ATP, but you've skipped 3 of the steps. When you absorb pantothene, you can go through step 1 instead of the enzyme pantothenic acid kinase, a very important enzyme in this pathway. Instead of converting pantothenic acid to 4-phosphopantothenic acid and then having to add the sulfur in 2 subsequent steps, converting it to 4-phosphopantothene, if you consume pantothene, that same enzyme will convert it to 4-phosphopantothene. Then you can skip 2 steps and again skip right ahead to the last 2 steps. So the rationale is that when you absorb pantothene and 4-phosphopantothene, you are skipping over many steps. You may be saving yourself from investing half the energy that you otherwise would. And if there are genetic variations in those enzymes, you'll be skipping over those enzymes. And so there might be a lot of people, again, we know so little about what increases our needs for B5. There might be a lot of people who have minor genetic variations in these enzymes who would benefit a lot by getting the preformed 4-phosphopantothene in or the more easily made coenzyme A from it. And I think that what can really illustrate this case is to look at a recently discovered set of genetic disorders in this pathway. So there are several inborn errors of coenzyme A metabolism that are mainly genetic disorders in the synthesis of coenzyme A. And the most common of these and the first discovered of these in 2001, was a recessive gene for a defect in the pantothenic kinase enzyme, the PANK. That's the first step in synthesis using pantothenic acid. This is known as pantothenic kinase associated neurodegeneration, or PCAN. It's estimated that 1 to 3 people out of every million people have PCAN. PCAN is separated into 2 types, the classic type and the atypical type. The classic type occurs early in childhood, and the atypical type is late onset. Although children can get the atypical type. Basically, the classical type is very, very severe, and that's why, on average, it happens a lot earlier and has a lot worse prognosis. Whereas the atypical type is a lot less severe, and so it tends to happen later, but it doesn't always happen late. And the the degeneration is a lot more slow, and the prognosis is a lot better. You're a lot more likely to live out a full life. In the classic form, it hits by age 6. In 90% of cases, it hits by age 6 in 90% of cases, and it can start as early as 6 months old or as late as 12 years old. It's marked by clumsiness and frequent falling, which are driven by rigidity and poor control over the muscles. It's frequently found with developmental delay, ADHD, and toe walking. Now, keep in mind that developmental delay, ADHD, and toe walking are a lot more common than this. So any of those things are not indications that it's probable that your child has this. So don't worry about that. But these patients, when they rarely live past the age of 20, the atypical form can hit as early as 1 year old, or it can hit as late as 37 years old, right? This is a genetic disorder that is weak enough that it may take you up to age 37. The average age of onset is 13. The overwhelming symptom, especially as it gets worse, is Parkinson-like akinesia. And the tremoring of the hands is less common, but it can happen. The posture is very unstable. And the typical slowness of movements and reduction of the size of movements that you see in Parkinson's happens to varying degrees. The atypical form also often is paired with OCD, impulse control disorders, anxiety, depression, and emotional lability. Think back to the quarrelsome nature of the people who were fed the antagonist. In the 1950s, when they induced B5 deficiency with an antagonist, the antagonist was an inhibitor of this enzyme, pantothenic kinase. So the pantothenic kinase deficiency is the late onset version bears a lot of parallels with what they had done in the experiments where they fed the antagonist. The main difference is that the pantothenic kinase enzyme has different forms, and the one that's affected in these genetic disorders is the one that dominates in the nervous system, and so it's much more focused in nervous system effects. Also, what you'll see in both the classic and the late onset versions are forceful contractions of the face or jaw or tongue in response to eating or speaking, and spasms that cause backward arching of the head, neck, or spine. Those are telltale signs that are more specific than OCD and impulse control, anxiety, depression, and emotional lability. The clinical guidelines recommend symptom management with all kinds of things and trying high-dose pantothenic, starting with 250 milligrams per day and increasing weekly by 500 milligrams per day until a dose of 2 to 5 grams per day is reached, or until side effects become evident. They recommend trying this for everyone who has it, but in general, it's the adults with the late onset version that report benefits. And that's because their enzyme is not completely defective. It's still able to use the high doses of pantothenate to some degree to compensate. But the children with the early onset form generally have difficulty taking the supplement, and they generally don't report the same kind of benefits because their enzyme is so defective that even throwing megadoses of pantothenate at it doesn't do anything. Now, researchers have tried to figure out what can we do about this that's better than just throwing megadoses of pantothenate at it. And they've developed mouse models to address it. And in the mouse, you don't have a perfect model because in the human, there's PANK1, which is the enzyme that's dominant in the liver, and PANK2 dominant in the nervous system. And the genetic defect is just in PANK2. And in the mice, it's not divided the same way. Their PANK1 is most abundant in liver, but their PANK2 is most abundant in their testes rather than their brain. So there's no specific way to target the defect right into the brain in mice like there is in humans. But as I mentioned before at the beginning, when the PANK2 defective mice are given a ketogenic diet, they get dramatically worse. They really start with devastating neurodegeneration. And pantothine supplementation, the form that Alex was talking about, can rescue them. Now, in humans, many people have tried a ketogenic diet, and in net, no one's reporting anything net positive or devastating or harmful from the ketogenic diet. So basically, ketogenic diets so far anecdotally don't seem to be doing anything one way or the other for this disorder. Also in humans, there are a lot of people trying Pantothenic supplements, but it's not clear whether they're working. There's no clinical trials to gauge the progress, and there's no overwhelming anecdotal support for using Pantothenic versus Pantothenic. However, one thing to note is that Pantothenic generates 2 molecules of Pantothenic when it's split, and Pantothenic does have some of the ability to bypass some of the steps and some of the ATP utilization that the 4-phosphopantothenic and the coenzyme A would in food. It's just that Pantothenic is not going to bypass anywhere near as much as you would bypass by getting 4-phosphopantothenic from food and from the digestion of coenzyme A in food. So researchers have continued to try to find things to do about this. And this was the basis for why in 2015 they found that 4-phosphopantothenic was stable in blood, and they tried it in mouse models and fruit fly models of the pantothenic kinase deficiency. And it works. The problem is that 4-phosphopantothenic is very difficult to synthesize in high amounts, in high purity, to give it to people as a high-dose supplement or as a drug, depending on how you would view supplementation with it. Also, it has a free sulfur group that makes it have a very bad shelf life. Now, if you compare that to the 4-phosphopantothenic in food, you don't have to synthesize it because the animals and plants are doing it for you. And you don't have to worry about the stability because it's bound up in the proteins in that food that protect it until you digest it. So when you're talking about food for phosphopantothenic, you don't have the stability issues that you have with developing it in pure form. So what they did was they developed an acetylated form of it to protect the sulfur from oxidation. And what they found was that in human embryonic kidney cells and in fruit fly larvae and in mice, when you made them genetically defective in pantothenic kinase, you could completely rescue the disease by feeding the acetylated form of 4-phosphopantothenic. Remember, 4-phosphopantothenic is one of the major forms in food. What's the other major form in food? Coenzyme A. What else can rescue fruit flies, larvae, live fruit flies, mice, zebrafish, and human cell models of pantothenic kinase deficiency? Coenzyme A. In every single model, preformed coenzyme A completely rescues the phenotype. The genetic disorder becomes not a genetic disorder when provided with enough 4-phosphopantothenic or coenzyme A, which are the 85% of the B5 found in food. And that makes you wonder what kind of doses do you need? Because as we'll talk about in part 2, there's not a lot of pantothenic in food, and really you're looking at nutritional yeast having the most and liver being a distant second and everything else being way below that. Which is funny, right? Because pantothenic acid is named after the fact that it is everywhere and in everything, but it's not very evenly distributed in the food supply. So I wonder what would happen with nutritional yeast supplementation and liver, eating a lot of liver, or at least managed, depending on the doses they need. You could overdose on the vitamin A or copper in liver. You could overdose on the selenium in nutritional yeast. But I do wonder whether the food forms of vitamin B5 from the foods that no one eats could have a significant benefit here. Now, there are also genetic defects in the final steps of coenzyme A synthesis and in some of the ones in the middle that are more recently discovered and that are more rarer. And for the final steps, none of the models have shown any benefit to coenzyme A or 4-phosphopantothenic. And the disease the way the disease manifests is very similar. So there are people with coenzyme A genetic defects who probably cannot be rescued by the supply of 4-phosphopantothene or any of these other things, although maybe certain drugs will be developed in order to help them. But where the benefit really is, where it's very promising, is in people with the pantothenic kinase deficiency. And so this has to make you wonder, what if there are polymorphisms that result in a 10 or 20% loss of pantothenic kinase activity? Could there be many of us out there who would be completely fine if we had enough food form vitamin B5, but who would suffer from at least moderate problems if we didn't, and who would not benefit from taking pantothenic acid supplements the way we would benefit from food form B5? I think you can make a very compelling case from this that consuming B5 in food is at least insurance against the possibility that you could have problems converting the pantothenic acid in supplements. And I think you could make a hypothesis that there may be polymorphisms out there in the world right now that are making some people benefit tremendously from eating the foods that are very rich in vitamin B5. So with that, this is the end of part 1, but we will come back for part 2 to talk about markers of nutritional status, how much we need, how to get it from food, and how to get it from supplements. This episode is brought to you by Ample. Ample is incredible. It's a meal in a bottle that takes a total of 2 minutes to prepare, consume, and clean up. 2 minutes. I'm not kidding. Now, I know what you're thinking. Anything that quick just has to be made of synthetic ingredients that you'd have a hard time pronouncing and wouldn't want to put into your body. But it's not. Ample is made entirely from natural ingredients and designed to provide an optimal balance between protein, fat, and carbs, as well as all the vitamins and minerals that you'd need in a single meal. There's no question that it's always best to sit down and take your time eating a home-cooked meal from fresh ingredients. But let's face it, oftentimes we just don't have time for that. If you live a busy life like I do and your goal is to get things done, you need quality fuel that you can get into your system quickly. Here's a great example where Ample is perfect for me. When I shoot videos, it takes hours to set up and break down all of my equipment. So I try to get as many videos shot in a day as possible. This prevents wasting a lot of time on setup and helps me conserve big blocks of time outside of shooting videos to get into a flow state where I can research something to my heart's content and spend all the time I need thinking about it creatively and analytically. But if I spend hours dealing with recording equipment plus hours spent preparing food, eating it, and cleaning up, there's no time left over to actually shoot any videos. So on recording days, I use Ample to maximize efficiency and focus on getting things done. Ample comes in 3 versions: original, keto, and vegan. And each version comes in 2 portion sizes, 400 calorie and 600 calorie. The 600 calorie original version gives me 37 grams of protein from a mix of grass-fed whey and collagen, which promotes satiety and flips my brain on. Its fat comes from coconut oil and macadamia nut oil. I like these oils because they're low in polyunsaturated fatty acids, or PUFAs, oils that promote aging and are usually loaded into the processed foods that most people eat when they need something on the go. The coconut oil provides some medium-chain fats to keep my energy levels up, too. The carbs, the vitamins, and the minerals all come exclusively from food sources like sweet potatoes, bananas, cocoa powder, wheat and barley grass, and chlorella. It's full of natural prebiotic fibers and probiotics to promote a healthy microbiome, and the gentle sweetness comes from a mix of honey, monk fruit, and stevia. I just mix it with water, drink it, rinse out the bottle, and boom, 2 minutes in, and I'm fully fueled and ready to face the next phase of the day. I first came across Ample when I met its founder and CEO, Connor Young, at PaleoFX a few years ago. Connor inspired me with his vision for Ample, which I anticipate will be much more than a meal in a bottle in the near future. I've become an official advisor to Ample, and I'll be helping Ample design scientific research that will lead both to an ever-improving Ample and, I hope, meaningful contributions to our understanding of how to use nutrition to help people be healthier and happier and perform better at the challenges that they care most about. As a listener to the Mastering Nutrition podcast, I've also worked out a special deal for you. If you use the discount code CHRIS15, you'll get 15% off your first order of Ample. To get your discount, go to amplemeal.com. That's amplemeal.com, 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 and use the code CHRIS15 at checkout. This episode is brought to you by Ancestral Supplements. Traditional peoples, Native Americans, and early ancestral healers believed that eating the organs from a healthy animal would strengthen and support the health of the corresponding organ of the individual. For example, the traditional way of treating a person with a weak heart was to feed the person the heart of a healthy animal. Modern science makes sense of this. Heart is uniquely rich in coenzyme Q10, which supports heart health. The importance of eating organs, though, is much broader than simply matching the organ you eat to the organ you want to nourish. For example, natives of the Arctic had very limited access to plant foods and got their vitamin C from adrenal glands. And vitamin C is important to far more parts of your body than simply your adrenals. In his epic work, Nutrition and Physical Degeneration, Weston Price recorded a story of natives who cured blindness using eyeballs, which are very rich in vitamin A. But now that we understand vitamin A, we know that we can get even more vitamin A by eating liver, making liver good for your eyes. Our ancestors made liberal use of organ meats both to be economical and to utilize their healing and nourishing properties. Animals in the wild do the same. Weston Price had also recorded a story of how the zoos in his era were capturing lions, tigers, and leopards, oh my, only to watch them become infertile in captivity. Researchers then observed what the lions did when they killed zebras in the wild. What they did was they went straight for the organs and bone marrow, leaving the muscle meat behind for the birds. But even the birds took what they could of the organs and bone marrow. Price reported that once the zookeepers started feeding the animals organ meats, boom, their fertility returned. The problem I often encounter, though, is that many people just don't like eating organ meats. Let's face it, if you weren't raised on them, it can be very hard to acquire a taste for them. That is where Ancestral comes in. Ancestral Supplements has a nose-to-tail product line of grass-fed liver, organ meats, living collagen, bone marrow, and more, all in the convenience of a gelatin capsule. For more information or to buy any of their products, go to ancestralsupplements.com. Ancestral Supplements, putting back in what the modern world has left out. This is part 1 of a 2-part series. Part 2 will be released in 2 weeks from today. If you would like part 2 now, you can get it right now by signing up for the CMJ Masterpass at chrismasterjohnphd.com/masterpass/mastering nutrition. Using that URL or the link in the description of this episode will earn you a lifetime discount of 10% off. You won't just get part 2 early, you'll get early content for all my audio video work. You get them as they are produced instead of as they're released to the public. I don't promise exactly when they come out, but that often means that you get them weeks or months in advance. You get the same benefits for Chris Masterjohn Lite, and not yet, but soon you'll be getting those benefits for Masterclass with Masterjohn. Chris Masterjohn Lite is short, quick, practical tips. Masterclass with Masterjohn is detailed biochemistry lessons as parts of a series that constitute a course on specific systems of the body. Finally, if you sign up for the Masterpass, you also get a at least monthly opportunity to participate in a live Zoom Q&A where you can ask me anything about nutrition. Or if I have a guest, you can ask us about anything about a specific topic that intersects with nutrition. If you want the other episodes in this series on managing nutritional status, you can get them at chrismasterjohnphd.com/marker episodes, or you can just go to chrismasterjohnphd.com/marker and scroll down to the episodes, or you can use the clickable link in the description of this episode. If you want an email update when the next episode comes out of Mastering Nutrition and when all the episodes of Mastering Nutrition come out, you can sign up at chrismasterjohnphd.com/mastering nutrition. If you want a dramatically more simple education on the nutrients than what you find in Mastering Nutrition, you can sign up for my beginner's course, Vitamins and Minerals 101, at chrismasterjohnphd.com/101. If you want to get all the super practical stuff about managing nutritional status for all the nutrients condensed down into one single comprehensive resource, You can get a copy of Testing Nutritional Status, the Ultimate Cheat Sheet at chrismasterjohnphd.com/cheat sheet. And you can use the code Mastering Nutrition to get $5 off. If you want more of my work in general, you can find me at chrismasterjohnphd.com. If you want more of Alex's work, you can find him at alexleaf.com. And you can find us on the internet. You can find both of us on Facebook, Instagram, and Twitter. You can find me on YouTube and every once in a while on Snapchat. All right, I hope you enjoyed this. Signing off, this is Chris Masterjohn of chrismasterjohnphd.com. This has been Mastering Nutrition, and I will see you in the next episode.